Welcome to the sixth lecture in the CSIR NET Life Sciences series by Utkalp Digital Classes. I am Richa, your mentor, and today we are going to deal with lysosomes. We are going to deal with the structure, functions, types of lysosomes, and the diseases associated with lysosomes. So, what exactly are lysosomes? Lysosomes are single membraned organelles found in cells. They form a part of the endomembrane system. Now, what is the endomembrane system? The endomembrane system, as we discussed in the last two lectures, is a system of continuous membranes which starts from the nucleus, goes on to the ER, Golgi, lysosomes, and so on. So all these organelles, the nucleus, ER, RER, SER, Golgi, lysosomes, all of these form a part of endomembrane system. So lysosomes were discovered by Christian de Douai. The size of lysosomes varies between 0.2 to 0.6 microns. They are often called suicidal bags. Why are they called suicidal bags? We're going to discuss in a bit more detail a little later. And the lysosomes contain transport proteins in their membranes. So what is the function of these transport proteins? These enable the lysosomal enzymes to be targeted into the lysosomal lumen. Now we are going to study the Gomoris reaction. Now it is a reaction which is a characteristic reaction for the identification of lysosomes. If we want to identify whether lysosomes are present in any particular tissue extract, we conduct this reaction or this experiment. So a tissue extract is taken, and that tissue extract is acidified with acetic acid to a pH of 5. So this pH 5 is really important for this reaction to take place. This solution, to this solution, beta glycerophosphate and lead ions are added. So beta glycerophosphate acts as the substrate. Now acid hydrolases from the lysosomes will cleave the phosphate group from beta glycerophosphate substrate. So this phosphate group will be cleaved by the acid hydrolases present in the lysosomes. All the lysosomal enzymes are nothing but acid hydrolases in nature. Now, so the cleaved phosphate group will combine with the lead ions to form lead phosphate. So this phosphate group will combine with these lead ions to form lead phosphate. And the lead phosphate which is formed is seen as a very dark blob under electron micrograph. So whenever this reaction is conducted and an electron micrograph is taken, if you see very large dark blobs, that is when we say lysosomes are present in the tissue extract. So lysosomal membrane has numerous V-type ATPases which are nothing but ATP dependent H+ pumps. So these proton pumps are responsible for acidification of the lysosomal lumen. So lysosomal lumen is made acidic to a pH of 4.6, and this pH is required. It is absolutely necessary for the function of acid hydrolases or lysosomal enzymes. So since the lysosomal lumen is acidic in nature and these enzymes function at an acidic pH, that is why these enzymes are called acid hydrolases. Hydrolases because they cleave, they hydrolyze. Now we are going to study how the lysosomal proteins which are synthesized in RER basically are sorted in Golgi and how are they delivered to the lysosomes. So once in the Golgi cisternae, these soluble lysosomal proteins are recognized for the addition of phosphate groups to mannose residues in the N-linked oligosaccharide chain. So phosphate group is added to the mannose residues of the N-linked oligosaccharide chain in the lysosomal proteins. We studied in the previous two lectures, in the RER lecture as well as the Golgi lecture, what N-linked oligosaccharides and what O-linked oligosaccharides are. These are the two types of glycosylation reactions which occur in ER and Golgi, wherein an oligosaccharide chain is attached to the newly synthesized proteins. So in N-linked glycosylation, the nitrogen of asparagine residues in proteins are attached to an oligosaccharide chain. So the lysosomal proteins which are N-linked, which are N-glycosylated basically, wherein the oligosaccharide chain is attached to the nitrogen of the asparagine residues of the protein. These oligosaccharide chains have mannose residues, and these mannose residues are phosphorylated in Golgi cisternae. Now, this mannose 6-phosphate tag acts as a sorting signal for lysosomal proteins. So once the mannose residues have been phosphorylated in Golgi, this mannose 6-phosphate tag acts as a sorting signal. So the phosphate is attached to the 6th carbon of mannose, that is why mannose 6-phosphate. Now this tag, what it does is, it is recognized by mannose 6-phosphate receptors in the trans-Golgi network. And once these mannose 6-phosphate receptors recognize this mannose 6-phosphate tag, this is when the entire protein is packaged into clathrin coated vesicles. So here you can have a look. This is the medial Golgi, wherein the mannose 6-phosphate tag is attached to the soluble lysosomal proteins. Towards the trans-Golgi network, you have receptors for mannose 6-phosphate tags, which are called as mannose 6-phosphate receptors. Now these receptors will recognize the mannose 6-phosphate tags on these proteins, and they will make these proteins be packaged into clathrin coated vesicles in the trans-Golgi network. Now these clathrin coated vesicles will have lysosomal proteins which are N-glycosylated and attached to mannose 6-phosphate tags. I hope you can understand till here. Now, these clathrin coated vesicles, they bud off, and as soon as they bud off from the trans-Golgi network, the clathrin coat is disassembled. Once the clathrin coat is disassembled, the mannose 6-phosphate receptors are also recycled back to the trans-Golgi network. So this says that when the clathrin coated vesicles bud off from the trans-Golgi network, they are into the cytoplasm, and now they are being transported to the lysosomes. In the cytoplasm, the clathrin coat will also disassemble, and the mannose 6-phosphate receptors will also be recycled to the trans-Golgi network, and ultimately these these proteins will further be targeted to the lysosomes. Now, the lysosomes contains a lot of enzymes, a lot of acid hydrolases, and these enzymes can be peptidases and proteases, nucleases, lipases, glycosidases, sulfatases, etc. Basically, all these are cleaving enzymes which function in the presence of an acidic pH. So most of the enzymes in late endosomes or vesicles budding from the trans-Golgi network are inactive zymogens. So all the vesicles, the clathrin coated vesicles which bud off from the trans-Golgi network, which contain the enzymes of the lysosomes, are actually, all these enzymes are actually proenzymes. They are not 100% active enzymes. They are synthesized and sorted in the Golgi as inactive zymogens. And when they fuse with the lysosomes, that is upon endosomal lysosomal fusion, these enzymes are activated by the pre-existing lysosomal enzymes. The pre-existing enzymes which are already present in the lysosomes will activate these zymogens into active enzymes. Now, why are these enzymes transported as zymogens and not as active lysosomal enzymes? So if they are matured and transported as active lysosomal enzymes from the trans-Golgi network itself, there is a possibility that these enzymes may end up reaching the cytoplasm and carrying out their functions in the cytoplasm and destroying food particles or other organelles, which is not required. So we require that these enzymes should function only upon the endosomal lysosomal fusion. These enzymes should function only when they are in the lysosome. So that is why they are synthesized as well as transported as proenzymes and not as active enzymes. Now, what are the functions of lysosomes? Now we are going to deal with the functions of lysosomes. The first function of lysosome is heterophagy. What do you mean by heterophagy? It is the digestion of materials of extracellular origin. So whatever materials lie outside the cell, that is the food or bacteria, they are engulfed by the lysosomes and they are digested by the enzymes present in the lysosomes. The second function of lysosomes is autophagy. Autophagy is nothing but the digestion of materials of intracellular origin. All the food particles or organelles or the molecules which are there inside the cytoplasm, the digestion of these materials is done by lysosomes, and this process is called autophagy. Auto means self, phagy means digestion. Now, the third function is autocytolysis, that is the release of hydrolases into the cytoplasm to digest the entire cell. So, suppose a cell is not functioning properly, or there is some problem in the biogenesis of the cell. That is when all the lysosomal enzymes will be released into the cytoplasm, and the entire contents of the cell will be digested by the acid hydrolases of the lysosome. And this process is called autocytolysis. Auto means self, cyto means cell, and lysis means breakdown. So self-cell breakdown is called as autocytolysis. So a very common example and a very important example is the breakdown of the tadpole's tail when developing into a frog. So when metamorphosis occurs in a tadpole, it has to transform into a frog. That is when it has to get rid of the tail. So a frog doesn't have a tail, but a tadpole has a tail. That is when autocytolysis will occur in the cells of the tail. The acid hydrolases from the lysosome will be released into the tail cells of the tadpole, and the tail cells will be digested. Ultimately, the entire tail will degrade and the tadpole will develop into a frog. Obviously, other mechanisms will also accompany the tail degradation. The next function of lysosomes is extracellular digest. So the hydrolases from the lysosomes are released outside the cell for the digestion of extracellular materials. Mind you, extracellular digest is different from heterophagy. Extracellular digest is when the components of the lysosome are released outside the cell. But in heterophagy, the components of the lysosomes, that is the acid hydrolases, are not necessarily released outside the cell, but they are released into the cell for the digestion of extracellular origin materials. But in extracellular digest, the acid hydrolases are compulsorily released outside the cell. So an example is release of enzymes by the sperm acrosome for the digestion of the egg's cell membrane. So whenever the sperm has to fertilize the egg, that is when the sperm releases the lysosomal enzymes for the degradation of the egg's cell membrane, so that all the sperm's contents can be released into the cell. So this is an example of extracellular digest. Coming on to the lysosomal diseases. These are very important from exam point of view. Questions will be directly asked on lysosomal diseases. So please study these diseases really well. The first disease is type 2 glycogenosis or type 2 glycogen storage disease. This disease is also called as the Pompe disease. It is an autosomal recessive metabolic disorder. What happens in this disease is it is caused by an accumulation of glycogen in the lysosomes due to the deficiency of the lysosome acid alpha glucosidase enzyme. So an enzyme of the lysosome, which is the alpha glucosidase enzyme, it is defective or it is absent from the lysosomes. So what happens is there is an accumulation of glycogen in the lysosomes. So this enzyme is responsible for glycogen degradation or conversion of glycogen to glucose. When this enzyme is inactive or absent, glycogen will not be converted to glucose and there will be an excess accumulation of glycogen in the cells. This damages the muscle and nerve cells throughout the body. So the muscle and nerve cells will have excess accumulation of glycogen. The second disease is the Hurler's syndrome. This is also known as mucopolysaccharidosis type 1 or gargoylism. It is a genetic disorder that results in the buildup of glycosaminoglycans or mucopolysaccharides in lysosomes. Now, these glycosaminoglycans are accumulated into the cell due to the deficiency or defect in an enzyme called as alpha L-iduronidase. It is an enzyme responsible for the breakdown of GAGs, the breakdown of glycosaminoglycans. So when this enzyme is deficient or the enzyme is defective, glycosaminoglycans tend to accumulate in the lysosomes. Now the symptoms include damage to the nervous system, skeletal system, eyes and the heart. Now one type of glycosaminoglycan whose buildup occurs in the body in this disease is dermatan sulfate, and another GAG is heparin sulfate. So dermatan sulfate and heparin sulfate accumulate in large quantities in the body in this disease. Now the third one is the Hunter's syndrome. This is also a very important lysosomal disease. It is also known as mucopolysaccharidosis type 2. It's an X-linked recessive disorder. This is caused by the deficiency of iduronase 2 sulfate. So this enzyme iduronase 2 sulfate is responsible for the breakdown of GAGs. Similar to the previous one in Hurler's syndrome, which is alpha L-iduronidase. Here it's iduronase 2 sulfate. This enzyme is responsible for the breakdown of GAGs. So in the deficiency of this enzyme, there is a buildup of dermatan sulfate and heparin sulfate in the body, and this damages the nervous system. So both Hurler syndrome and Hunter syndrome are almost similar. The only difference is in the enzyme. Coming to Taysacs disease, there is an accumulation of gangliosides and other lipids in the nerve cells due to the absence of the enzyme beta-hexosaminidase A. Taysacs disease has come in the exam a lot of times. Sometimes they will ask the enzyme responsible for the Taysacs disease. Sometimes they are going to ask what happens, like what accumulates in when there is a Taysacs disease condition. So you need to be very thorough with this enzyme and the symptoms, the accumulation of gangliosides, and what is the effect of Taysacs disease, and you have to go through this thoroughly. So there is an accumulation of gangliosides and other lipids in nerve cells. This is also very important. This occurs in the nerve cells, and this is because of the deficiency of beta-hexosaminidase A enzyme in the lysosomes. Affected children get paralyzed within 6 months of birth and most die at the age of 6. So this is a fatal disease. Next is the eye cell disease. The enzyme deficient or absent is N-acetylglucosamine phosphotransferase. This is a cis-Golgi enzyme. Now, this enzyme in the cis-Golgi, when this is absent or deficient, the mannose-6-phosphate tag cannot be added to the N-linked oligosaccharide, the protein which is N-linked N-glycosylated. Mannose-6-phosphate tag cannot be added to the lysosomal protein. So when mannose-6-phosphate tag cannot be added, what will happen? The mannose-6-phosphate receptor will never recognize that protein as a lysosomal protein, and that protein will never be exported out of Golgi and it will never reach the lysosome. So the lysosomes will be devoid of any enzymes or proteins and the lysosomes will be called as empty ghosts. So basically there will be no enzymes in the lysosomes. The lysosome will be dysfunctional, and this is due to the deficiency or absence of N-acetylglucosamine phosphotransferase. Now the last disease is Gauchers disease. There is an accumulation of glucocerebrosides in lysosomes and macrophages due to the absence of glucocerebrosidase. So this enzyme is responsible for the breakdown of glucocerebrosides in lysosomes and macrophages. Since this enzyme is absent or deficient, glucocerebrosides accumulate in the lysosomes and macrophages. This usually affects children. Symptoms include enlarged liver and spleen, anemia, and mental retardation. So with the lysosomal diseases, we've come to the end of lysosomes. In the next lecture, I'm going to deal with peroxisomes, glyoxysomes, and in the later lectures, we'll be dealing with mitochondria, plastids, and plasma membrane. Thank you.